the use of absorption enhancers and all these things. If you have any kind of quantification of these parameters regarding the bioavailability, in addition to some in vivo tests.
Yeah, sure.
We have a systematic way to address this kind of issue instead of just knowing that there are some effects.
Sure. Yeah, no, I think it's a good comment. So I've not shown it here because for the sake of time, but of course the selection of these absorption enhancers is based on a lot of first in vitro studies, then animal studies. They are not very good models, I would say. You can get a very high bioavailability in rats, more than 10%. Then in dogs, it will go to a few percent, and in humans, less than 1%. It's a typical range. But even if you go to less than 1%, it's much more than without a permission enhancer where it's totally flat. You don't have any absorption at all. So the permission enhancer really brings something. It's shown. I would say the mechanisms are not well known today. People claim it goes transcellular or paracellular, but well, there's not so many convincing data in the literature, I would say.
Okay, thank you. Thank you.
One more. Thank you. Okay. And you said the PEG conjugate they used for the oral peptide. Okay. What's the mechanisms, and also you have evidence of that?
Can you repeat this?
The mechanism of the PEG conjugate for the oral delivery.
Yeah. So there are companies working on PEG conjugation. So, I would say PEG might be interesting in mitigating efflux PGP effects. It has been demonstrated. I've not seen so many products in the clinics right now. There are some nanoparticles also using some PEGylated systems.